A very good afternoon to one and all. In the previous class, we have discussed about uh, basics unit 1, unit 2, that means block 1, block 2. Uh, we have discussed introduction to immunology or immune system, and the lymphoid system, okay, cells involved in immune response, and we have also discussed about humoral immunity and cell-mediated immunity in brief. Today, I would like to uh, tell you about structure and functions of immunoglobulin. Okay, before going to introduce about immunoglobulin, I would like to tell you about antigens. Okay, antigens are the molecules or the substances uh, or is a substance foreign to our body that evokes an immune response is called as antigen. Our antigens are molecules that stimulate the immune response called as immunogen or antigen, because uh, these antigens are maybe proteins, maybe polysaccharides or glycoproteins or lipoproteins. Different molecules are act as a uh, antigen. Those are having the capacity to evoke or stimulate our immune response or immune system or immune response. And these are antigens are two types. Those are the exogenous antigen and endogenous antigen. Okay, exogenous antigen and endogenous antigens are there. And in this chapter, I would like to uh, describe about immunogenicity. Okay, the immune response which we will get against the immunogen by immune system is called as immunogenicity. What is immunogenicity? It is the immune response which we will get against the immunogen or antigen by immune system. Whenever the immunogen or antigen encounters against our immune system, we will get this immunogenicity. After fighting against the antigens, our body gets immunogenicity. And this is individual specific and antigenic specific. So we have another terminology, that is epitopes. Before going to know about antibody, you must know about these terminologies. What is antigen or immunogen? What is immunogenicity? And what is epitope? What are the adjuvants? What is paratope? You must know about these things before going to know about antibodies. So epitopes. What is epitope? Immunologically active region. Epitope is the immunologically active region of immunogen. See, immunogen is a large particle or large molecule on which this epitope is there. Okay, this area is immunologically active region on the immunogen. The binding site for the antibody is also called as epitope. You can describe in this way also, whether that meaning or whether that or this. Immunologically active region, you can say, or binding site for the antibody on the binding site for the antigen on the antibody. Okay, uh, epitope binds with antibody, lymphocytes, macrophages. Okay, it can bind with epitope can be bind with the antibody binding efficiency with the antibody. It also having the binding efficiency with the lymphocytes and macrophages. These are the three molecules, uh, binding molecules for the antigen. Okay, antigen goes and binds with these three molecules. Antigen determinate site and the site at which immune cells will bind. Okay, the site at which immune cell will bind. We can also define in this way also. The site epitope is the site at which immune cell means antibody, immune cell or antibody. You can understand in these two ways. The site at which immune cell or antibody will bind. Okay, the site can also be called as epitope. And coming to the haptens, okay, hapten is a molecule that reacts with antibody and immune cells and individually cannot elicit the immunogenicity. Hapten itself cannot elicit the immunogenicity, but and detected by antibody and immune cells, but cannot generate immunogenicity. It can detected by the antibodies or and immune cells, but cannot generate immunogenicity like antigen. And incomplete we can also call as incomplete antigen. Happens also called as incomplete antigen. And these are the small molecules, and these are actively provoke immunogenicity when they are combined with carrier proteins. Okay, yeah, along with the carrier proteins, it can elicit the our immune system or immune response. Uh, Alone, it cannot generate or it cannot provoke, it cannot evoke our immune system. But whenever these haptens combined with the carrier molecule, along with the carrier molecule, it can evoke or stimulate our immune system. And it can be foreign substance, a non-protein substances, low molecular weight haptens characteristics, and itself cannot generate an immune response. By activating, by attaching with the carrier, it can generate immune response. These are the characteristic features of hapten. Okay, hapten is a off antigen. We can call it a half antigen or incomplete antigen. In combination with carrier molecules, it can generate uh, immunogenicity in the our system. And coming to the adjuvants, adjuvant adjuvants are the substances that are not immunogenic but increase the immunogenicity. Okay, these are the not immunogenic molecules, but it increases the immunogenicity of uh, added immunogen. See, it is individually it cannot elicit the acts as a immunogen, but when it attaches to the antigen or immunogen, it enhances the property or action of the antigen. Whenever these adjuvants are added to the immunogen or antigens, the property of immunogen or antigen will be enhanced after adding adjuvants. Okay, and protects the antigen, protects the integrity of the antigen. It protects the integrity of the antigen and targets the antigen presenting cell. Okay, induces the cytotoxic lymphocytes, non-living vaccines. Weak antigens are example for the adjuvants. And whenever the weak antigens combined with the adjuvants, it elicits the immune response. Okay, aluminum hydroxide example for the adjuvant is are aluminum hydroxide, aluminum phosphate, calcium phosphate. Egg albumin sometimes also acts as a adjuvant. Okay, these are the molecules or compounds. Whenever these molecules are added with the weak antigen, like uh, a non-living vaccine, we can call it as weak antigens. Whenever these adjuvants are added to the non-living vaccine, okay, the immunogenic property of the weak antigens will be enhanced after adding these adjuvants. Okay, and coming to the paratube. Okay, paratube. We will discuss this about this terminology in the structure of antibody. So you have learnt about antigen. Okay, immunogenicity, epitope, and hapten. adjuvants. Okay, we will discuss now. We will discuss about antibodies. Okay, what are the antibodies and what is the structure of antibody? We will discuss uh, clearly. Okay, and antibodies are the globular proteins. I have already told you in the previous class. These are the also called as magic bullets of immune system. Okay, magic bullets of immune system is bullets of immune system are antibodies. Okay, these are the glycoproteins or globular proteins secreted or synthesized against an antigen from effector B cell, that is plasma cell. Okay, where exactly from these molecules are secreting from activated or effector B cell, that is plasma cell. One is that memory cell, another one is a plasma cell. Plasma cell secretes the antibodies. Okay, and based on the mobility, antibodies. I have already told you that based on the mobility, that there are two types of antibodies are there. First one is circulating antibody, and the next one is surface antibodies. Okay, and coming to the structure, this resembles as a Y-shaped structure. Okay, it resembles as Y-shape. Okay, and it consists four polypeptide chains. Okay, four polypeptide chains are there. What are they? Two heavy polypeptide chains, two light polypeptide chains. Two chains are linked with covalent disulfide bonds. Okay, disulfide bonds helps to connect with each other. Those two polypeptide chains and heavy chain variables are alpha, gamma, mu, and delta, epsilon are variable varieties of heavy chains are there. And light chain variables are kappa chain, lambda chain. Chain names are there for the heavy chain and light chain. Two ends of uh, antibodies are amine terminal and carboxylic terminal. Okay, and it has two regions. Okay, it has two regions. One is that fab region. Okay, one is that fab region, and this is the fab region, and uh, from here to here, this is the fab region, and uh, mm, from here to there, that is FC region. What is fab region? Fragment antigen binding site. Whenever the antigen come into the system, this antigen processed by the different cells, and this antigen goes and sits on this antigen binding site. Okay, that is one of the fragment or the region. And in another side, it contains FC region. FC region is a uh, uh, means FC means fragment crystallizable. Okay, fragment crystallizable or fragment cell binding region. Okay, this is a FC region with which antibody binds with the cells. Okay, immune cells like macrophage and neutrophil and eosinophils, mast cells, basophils, and B cells. Okay, different types of uh, cells can be the FC region can be bind with the different types of FC receptors on the surface of the different cells, like macrophage, dendritic cells, neutrophils, natural killer cell. Okay, natural killer cell also having the FC region for different types of antibodies, and eosinophils, mast cell. Okay, mast cell, basophils. These are the cells are having the which are having FC region receptor on its surface. So this antibody goes and binds with those FC receptors with the help of FC region. This is about the FC region. Whenever this FC region goes and binds with the FC receptors on the surface, biological cascadial reactions will be stimulated or activated by the responding by the giving the signal from the uh, antigen antibody, action, and further subsequent reactions may be leads to the different consequences. So uh, and it also contains domains. Okay, heavy chain, that means domains means folded areas. Okay, I will show where exactly domains are there. Hmm. Mhm. Domains means these are the folded areas. Okay, folded areas are there. One fold is one domain. Another fold is another domain. And VH means variable. Okay, variable region. C means constant region. Okay, variable light chain. VH means variable heavy chain region domain. VH means variable heavy chain domain. CH means constant heavy chain domain. Variable VL means variable light chain domain. CL means constant light chain domain. Okay, these are the terminologies. And CH2 means constant heavy chain domain. Constant heavy chain domain three. Okay, three uh, constant domains and one heavy chain domain. Variable domain. There are four heavy chain domains are there. One is VH and three another three are the constant heavy chain region domains. Light chain, coming to the light chain, there are two domains are there. One is light chain, VL, CL, enough. And hinge region is they also be there in between two heavy chains. This is the hinge region, which is connected by disulfide bonds. And this is hinge region can be movable. It can be movable with the help of hinge region. And light chain, coming to the light chain, smaller polypeptide chain, molecular weight 23,000 Dalton or 23 kilodalton. L chains are two types. One is kappa chain, another one is lambda chain. Okay, and kappa chains are four types in human. Okay, kappa chain is only one type. This is about the light chain. And heavy chain, coming to the heavy chain, is the longer polypeptide chain, three times larger than light chain. Three times larger than light chain. Molecular weight 50,000. Molecular weight 50,000 to 70,000 Daltons. Okay, and heavy chains are five types. Okay, based on the molecular structure, heavy chains are five types. What are they? Gamma chain, mu chain, alpha chain, epsilon chain, delta chain. Based upon these chains, the scientist named categorized into five types. Antibodies are categorized into five types based upon the presence of heavy chain. Which type of heavy chain is there in the antibody? They have categorized into five types. One is that immunoglobulin G, which is having gamma chain in its has a heavy chain. Immunoglobulin M is the second type. Immunoglobulin A contains alpha chain. Immunoglobulin E contains epsilon chain. Immunoglobulin D contains delta chain. Okay. In the along with these chains, in the short or light chain, both type of whether kappa will whether the kappa will be there or delta will be there, lambda will be there. Okay. Not is the rule is not for the light chain, but rule is there for the uh, heavy chain. Okay, if you are talking about immunoglobulin G, the compulsory it contains gamma chain. Okay, that is the rule. This is about the basic structure of uh, this antibody. Okay, yeah, I have discussed about functions of antibody, but today I would like to discuss about types of different classes of antibodies. So we have, as we have discussed earlier, the types of antibodies uh, based upon the heavy chain. One, two, three, four, five types of uh, antibodies are there. They coming to the structure of different types of antibodies. First of all, immunoglobulin A. Subtypes are Immunoglobulin A is only one of the subtype. Okay, and especially in depth, it contains two subtypes, alpha 1 and alpha 2, means immunoglobulin class 1, subclass 1, immunoglobulin A subclass 2. It contains two subclasses. Immunoglobulin A contains two subclasses, and secreted from it is where exactly these immunoglobulins are secreted. Mainly dimer, also monomer, trimer. Sometimes secreted from exactly plasma cell. Okay, these are the secretory uh, immunoglobulins. Okay, functions I will explain in detail uh, about this function, immunoglobulin classes in detail. But just I would like to mention structure, whether these molecules are monomers or dimers or pentamers. Just for this point, I have I will display. I have displayed this image for the purpose only. Immunoglobulin A is the dimer. Okay, sometimes trimer also be there, but immunoglobulin D always is monomer, and coming to the immunoglobulin E is also monomer. What we have discussed the structure exactly, one monomer. Okay, it when it attached to the another with the help of J chain, that is a dimer. If the three antibodies, normal antibodies attached together, that is trimer. If five antibodies, normal antibodies attached together with the help of J chain, that is pentamer. In case of immunoglobulin G, it also be the monomer. Immunoglobulin M, it is the pentamer largest immunoglobulin. And coming to the another points regarding the antibody, yes, one by one we will study each and every point about immunoglobulins. So I would like to start from immunoglobulin G. Immunoglobulin G, just wait for a minute. I will. Reserve. Yes, yes, yes. Immunoglobulin G is the most abundant in the blood or serum. Is the most immunoglobulin G is the most abundant immunoglobulin among the all. It is the percentage is that 80 percentage. Okay, 80 percentage of the among the 100 percentage, 80 percentage of the antibodies are immunoglobulin G, because that's why we are calling as most abundant antibody in the blood or serum, and provide bulk immunity against blood-borne infection. Half-life of the antibody 7 to 23 days. Okay, monomer having two epitopic binding sites. Okay, epitopic binding sites means uh, antigen binding. Okay, antigen binding site. This is one antigen binding site, and another one is this. So the immunoglobulin G can bind with two antigens. Okay, it can bind with two antigens. That's why antigen binding number of antigen binding sites are two, or we can also call as two epitopic binding sites. The smallest antibody, the another one important characteristic feature is that the smallest antibody is the immunoglobulin G. That's why it's crossing mother placenta and entering into the fetus. Okay, and the only one antibody which can cross the placenta, main functions of coming to the functions of immunoglobulin G is that it is the only one antibody which can cross the placenta because of its uh, small uh, structure. It will give immunity to newborn infant. Okay, because uh, after birth, from 2 to 3 months, there is no B cells maturation will not be there after 2 3 months, and so any antibodies, no antibodies are produced in the newborn children or newborn babies. So it will give immunity to newborn infants against different types of pathogens and parasites, and neutralizes the toxins in the always, not in children, but all stages. It neutralizes the toxin. First step, it neutralizes the toxin. Immunoglobulin subclasses are there. Subclasses one, subclass one, two, three. Okay, four types of subclasses are there for the immunoglobulin G. Especially immunoglobulin G3 binds to FC receptors of phagocytes. Very important point is that, especially among the four subclasses, immunoglobulin 3, the immunoglobulin 3 uh, having the FC receptor of phagocyte. Okay, it is a FC receptor and binds with the FC receptor of phagocyte. It is the FC receptor, the FC region of the this G3 antibody can bind with the phagocyte cells. Okay, especially immunoglobulin G3 class subclass. Okay, and most bulk immunity will give bulk immunity, crosses the placenta. You know about these points. This is about immunoglobulin G. Okay, and coming to the immunoglobulin A, uh, heavy chain is alpha chain. You know, among the 100 percentage, among this 6 percentage of the immunoglobulin A will be seen, can be seen in the body or blood or serum. They found mainly in the body secretion like saliva, okay, mucus, tears, colostrum. Colostrum means first milk, first mother milk. We can call as colostrum. After that milk, we can call it as milk. All these secretions contains abundant of immunoglobulin A. So we can also call as secretory immunoglobulin. Immunoglobulin A also called as secretory immunoglobulin. It can bind with four epitopes. It has capacity to bind with four antigens at the same time. So secreted from plasma cells of mammary glands, salivary glands, okay, respiration, gastrointestinal tract, okay, vaginal tract, etc., etc. From where exactly these antibodies are produced, protected the gut pathogens of neonates. Okay, protect the gut pathogens of neonates. In the milk, it contains immunoglobulin A. That's why it protects babies in gastrointestinal tract against the different types of infections. This is about the immunoglobulin A. Coming to the immunoglobulin M, and the largest immunoglobulin among the all is immunoglobulin M. Immunoglobulin M is the largest immunoglobulin among the all, and pentameric in structure. Okay, pentameric in structure, as I have told you earlier, and 10 epitopic binding sites are there in the immunoglobulin M. Okay, at the same time, it can bind with the 10 antigens. Okay, and that is the beauty of immunoglobulin M. And half life is 5 days. So don't bother about half life. And functions. Coming to the functions, very important. First immunoglobulin class produced, not provided. First immunoglobulin class produced for foreign particles as the primary response. Okay, non-specific antibodies. The first immunoglobulin class produced for foreign invaders. This is first line or first immunoglobulin are produced in our body, and especially ABO blood group antibodies also act as a ABO antibodies. These are immunoglobulin M type, belongs to the blood groups. Okay, on the surface of blood cells, red blood cell antibodies. We are saying antibodies are there on the red blood cell. Those antibodies are especially immunoglobulin M type, and give the defense against the multivalent antigens, act as opsonin. Okay, wherever the opsonin term is there, there is a role. There is a role, key role of the immunoglobulin M is there. In the previous picture, I have not mentioned about classical uh, pathway system. Just wait for a minute. Okay. And these antibodies also having uh, the site for the complement protein binding. Okay, where exactly they are there, and here uh, some of these region having the complement binding site. Complement binding site means complement protein come and bind with this site, and this binding will facilitate the opsonization signal and will give the opsonization. It means it enhances the phagocytic pathway. Okay, phagocytosis process. Wherever the opsonization is there, opsonization leads to the phagocytosis pathway. Okay, phagocytosis process. In this, immunoglobulin M play a role in the opsonization. By it may leads to the phagocytosis. And coming to the immunoglobulin D, uh, this is one of the monomer containing antibody. Lowest quantity is there in our body or blood system. Best example for the surface antibody on the B cell. It is one of the surface antibody, and most of the times the functions of the immunoglobulin D not yet clearly known. So don't bother about immunoglobulin D. Just remember that this is the surface antibody. Okay, best example for the surface antibody. And coming to the immunoglobulin E, the heavy chain is that epsilon chain, and a very low levels of immunoglobulin is there in our body. Significant role in enhancing allergy, acute inflammation. It play a key role in enhancing the allergic reaction. Where we have talked about this immunoglobulin E in the hypersensitivity reaction 1. Okay, it play a key role in the allergy making reactions, in the inflammatory reactions. Wherever you are, wherever the inflammation is there, wherever the allergic reactions are there, the role of the immunoglobulin is the necessary. Okay, very important. And allergic reactions we will find in the allergic reactions we will find immunoglobulin E. Okay, in more number. Okay, after allergy, after testing where the percentage of the immunoglobulin E is more in those allergic area region. And most cell membrane is having mast cell. Mast cell membrane is having the receptor for the immunoglobulin E, that is FC receptor. The name of the receptor is that FC
Hello?
R1. Excuse me. Tell me, Amma. Hello? Hello?
Sir?
Hello? Am I audible? Hello, Amma. Hello? Hello? Hello?
Hello, sir?
Amma, am I audible, Amma?
Yes, sir.
Tell me, Amma.
సార్ ఇప్పుడు
Can you
ఎవరికైనా
know?
దురదలు వస్తున్నాయి బాగా అంటే అది ఐజీఈ దీని వల్లనా సార్?
ఐజీఈ వల్ల కదమ్మా, ఏదైనా మనకు అలర్జిక్ ఏజెంట్ లోపలికి ఎంటర్ అవుతుంది కదా, అది మన యాంటీజెన్ గా పనిచేస్తుంది అనమాట. మనకు
అంటే మీరు
ఫారెన్ పార్ట్ ఆ యాంటీజెన్స్ మన లోపలికి ఎంటర్ అయిన తర్వాత మన బాడీ రిలీజ్ చేసే యాంటీబాడీస్ ఏమిటంటే ఇమ్యునోగ్లోబులిన్ ఈ టైప్. అక్కడ ఎందుకు ఇమ్యునోగ్లోబులిన్ ఈ వస్తుంది అంటే
ఓకే సార్.
దాని వల్లనే అలర్జీ వస్తుంది అనమాట మనకు. ఎందుకు అంటే ఇమ్యునోగ్లోబులిన్ ఈ అనేది ఆ మాస్ సెల్ పైన అటాచ్ అయినప్పుడు ఆ బర్స్ట్ అవుతుంది, డీగ్రానులేషన్ జరుగుతుంది. దాని గ్రాన్యుల్స్ నుంచి రిలీజ్ అయిన కెమికల్స్ అక్కడ స్ప్రెడ్ కావడం వల్ల
ఓకే.
ఆ ఏరియాలో మంట రావడం, దురద రావడం, ఓకే, స్వాల్ కావడం, స్వెలన్ కావడం, పెయిన్ రావడం అనేది జరుగుతుంది. దాన్ని మనం అలర్జీ అని చెప్తాం.
ఉమ్.
అలర్జీ మేకింగ్ యాంటీబాడీ అంటే మనం
ఓకే సార్.
ఇమ్యునోగ్లోబులిన్ అని చెప్పొచ్చు. ఇమ్యునోగ్లోబులిన్ ఈ వల్ల మనకు అలర్జీ రాదు. ఎప్పుడైతే యాంటీజెన్ వస్తుందో అప్పుడు యాంటీజెన్ వల్లనే అలర్జీ వస్తుంది. ఆ యాంటీజెన్ ని మనం యాంటీజెన్
ఓకే.
నుంచి ప్రొటెక్ట్ చేయడానికి మన ఇమ్యునోగ్లోబులిన్ ఈ ప్రొడ్యూస్ అవుతుంది అన్నమాట.
ఓకే. ఓ-
అర్థమైందమ్మా?
ఓకే సార్.
ఓకే. సో this is about this also play a key role in type 1 hypersensitivity reaction. Okay, what we have explained, what we have discussed about this type 1 hypersensitivity reaction. Especially in this reaction, immunoglobulin E play a key role uh, to making the hypersensitivity reactions reaction 1. So this is about the different types of antibodies. Okay, antibodies structure, antibody subclasses, classes, subclasses, and functions of different types of antibodies. So, coming to the function, overall, these antibodies are uh, neutralizes the different types of antigens. Okay, first of all, the first function is of the antibody is that neutralization. Neutralization means it goes and binds with the epitope. Okay, assume that this is the antigen, and this is the epitope exactly. Okay, this is the epitope, and this antibody go and bind with this epitope region, and it neutralizes it. It silences the uh, activity of antigen, especially epitope region. Okay, it furthermore indirectly antigen is inactivated. Okay, antigen is inactivated, get inactivated after attaching with the epitope region with the antibody. ఎప్పుడైతే యాంటీబాడీ అటాచ్ అవుతుందో అప్పుడు ఈ యొక్క యాంటీజెన్ అనేది సైలెన్స్ అయిపోతుంది. అది ఆ యాక్టివ్ సైట్ అనేది క్లోజ్ చేయబడుతుంది కాబట్టి అది మనకు రియాక్షన్స్ అనేవి ప్లే కావు. సో న్యూట్రలైజేషన్ ఈజ్ ద ఫస్ట్ ఫంక్షన్ ఆఫ్ ది యాంటీబాడీ, అండ్ సెకండ్ ఫంక్షన్ ఈజ్ దట్ lysis. Okay, lysis means complement activation. Okay, in just before I have told you that it also having in the Fc region of the antibody also having complement protein binding site. Okay, this is a complement protein binding site, and uh, in other at the region, this is the antigen binding site. Whenever these antigen antibody complexes are there in our body, and this complement binding site will be opened for the binding of the complement protein, and this complement protein go and bind with this site, and this binding facilitates the activation of different types of complement pathways, classical pathway or alternate pathway, lectin pathway. Different types of pathways are activated after getting interacted with the uh, antibody uh, receptor. Okay, antibody receptor with the uh, protein, complement protein. Whenever this complement protein go and bind with the this receptor of the on the antibody, and this may call the different types of uh, cells and different types of subunits of the complement proteins. What we have talked about the complement protein system, complement system in the alternate pathway, in the classical pathway, in the lectin pathway, and leads to the different immunological responses. One is that complement activation, by which the membrane attacking complex will be formed. After that, on the surface of the foreign body or infected cell, pores will be formed. By after pore formation, the contents of the cytoplasm will be used as out. After this, the cell death will be the consequence. Okay, this is the lytic function of the antibody. And another function is that opsonization. Just I have told you that what is opsonization. The opsonization is that it attracts this thing. Okay, by this pathway, it activates the phagocytosis. Okay, it attracts the different type of phagocytic cells to the site. Whenever this antibody binds with the complement protein, not only leads to the glytic pathway, but also leads to the phagocytosis, but also attracts the phagocytic cells towards the infected site. The binding of complement protein with the complement receptor on the surface of antibody or Fc region, it attracts the different types of phagocytic cells to the uh, infection site, okay, to the infected site or different sites where exactly required. Requirement of the phagocytosis, phagocytic cells are there. So this is the major function. These are the three the major functions for the immunoglobulin. If you have any doubts on the immunoglobulin, you can ask. No, any doubts అమ్మా ఏమైనా డౌట్స్ ఉన్నాయా ఇమ్యునోగ్లోబులిన్ గురించి?
ఏం లేవు సార్.
So now I would like to talk about major histocompatibility complexes. Okay, major histocompatibility complexes.
ఓకే సార్.
In the second unit block, we have talked about, we have discussed about humoral immunity in brief. As a unit 6, unit 7 also uh, discussed in brief, that is cell mediated immunity. We in this block, two chapters are left, uh, unit 5 and unit 8. Okay, these two topics will be discussed today session. So coming to the major histocompatibility complexes. Major histocompatibility complexes are the large genomic regions from where chromosome 6 in the human beings, chromosome 6 uh, covers, produces this codes, encodes this major histocompatibility complex. What is major histocompatibility complex? This is the region or large genomic region containing a cluster of genes encoding a cell surface molecules. Okay, major histocompatibility complex means large genomic region in the our DNA. Okay, and it contains a cluster of genes encoding cell surface molecules. Whenever this MSC genome is get activated, and it gives MSC molecule and which expresses on the molecule on the surface of the cell, infected cell or uh, invaded, okay, invaded cell. Okay, that is the major histocompatibility complexes. Okay, and are required for antigen presentation. And very important point is that these are the required molecules for antigen presentation to T cells, especially and for the rapid graft rejection. Okay, for the rapid graft rejection or for the immunological response, these are the molecules are required for T cells, not other cells recognize as the MSC molecules, but only T cells are having the capacity to recognize as or study the information or taking the information from the MSC molecule, because it having the it expresses along with the antigen after getting activated by the transcription pathway, and it uh, takes the antigen after production, and it expresses on the surface along with the antigens. So MSC was uh, the response was elicited because the host recognized the major histocompatible molecules on each cell of the graft. You don't bother about the graft, and in the MSC, regarding the MSC molecules are the large genomic complexes with multiple loci. Traditionally, it is divided into three regions. Okay, MSC molecule 1, class 1, class 2, class 3. And MSC molecules also occur in the group of vertebrates. Okay, all vertebrates contain this type of MSC molecule. MSC is the generic name. Okay, the collective name given to antigen produced by MSC varies for different species. In humans, it is called as HLA complex. That means human leukocyte antigen. Human leukocyte antigen is the name of the MSC in the human being, and the name of the MSC molecule in the mice is that S2 complex. Name of the MSC molecule in the dogs is called as dog leukocyte antigen. Rat leukocyte antigen is rabbit. Rabbit leukocyte antigen in the rabbit, and guinea uh, pig leukocyte antigen in the guinea pig. And based upon the species of the organism or vertebrate, the name is differs from each other. And it is the large molecule can be divided into three regions or three classes. Class 1, class 2, class 3 are the major types. Coming to the class 1 and encoded, I will show one diagram for you. Just wait for a minute. All the cells contains MHC molecule 1. I will show for your reference. I will show one uh, image. Just wait for a minute. MHC. Yeah. Okay. Yes. So these are the MSC molecules, especially three types. But in your for your understanding or for your stage MSC stage, MSC period, MSC one, MSC two is enough for study. Structure, coming to the structure, alpha chain. The MSC one contains alpha chain, okay, and beta B2 chain, B2 chain and alpha chain. MSC two contains alpha chain and B chain. Okay, two domains in the B chain, and in for MHC1, three domains in the alpha chain. In cell distribution of the MHC1 molecule, surface of all nucleated cells contain MHC1 molecule. Okay, all nucleated body cells are having this MHC1, not MHC2. But especially MHC2 molecules are there only in few of the cells, especially which are active in the immune system or immune response. Those cells are called as antigen presenting cells. Okay, or these are the two types: professional antigen presenting cells and non-professional antigen presenting cells. And on the surface of these antigen presenting cells, this MSC molecule 2 is there. Okay, and present antigens, cytotoxic T cell, always MSC1 molecule, MSC1 uh, molecule complex present it antigens to the cytotoxic T cell, and wherever the MHC molecule 2 expresses on the surface of the cell, that is antigen presenting cells, it submit the it present the antigens to the helper cell, T helper cell always. And binding site for the T cell is that CD8 marker. Clustral differentiation site, 8 receptors on the cytotoxic cell binds with the MHC molecule always. And coming to the MSC molecule 2, CD4 receptors on the surface of T helper cell binds with the MSC molecule 2 always. There is a rule. Bind with the endogenous antigen. Okay, whenever the endogenous antigens are there, are produced in our body, that especially bacteria, viruses. Okay, endogenous means toxins of the bacteria, toxins of the virus uh, will be act as endogenous antigens. At that time, these antigens expresses on the surface of the infected cell with the help of MHC molecule 1 always. And exogenous antigens can be processed and expressed with the help of MSC2, presented, especially presented by the MSC2 molecules. And human nomenclature for the MSC molecule is that human leukocyte, antigen A, subclass A, antigen A, subclass B, subclass C. These are the three classes, the classes of the human MSC molecule 1 is there. And for the MSC molecule 2, DR, DQ, DP, three types of MSC molecules are also there. And mouse, we can also, I have also mentioned that in the mouse, we can call it as H2 complexes. In the human being, HLA complex. In the mouse, in the mice or mouse, we can call it as H2 complex. These three subtypes are there: 2K, 2D, uh, 2L. So these are the for the MSC molecule 1. For the 2A, E, two subtypes are there. Don't bother about genes and gene sequences, different phenomenon, and just remember two things: MSC molecule 1 structure, MSC molecule 2 structure. MSC molecule 1 uh, presents the antigens always to the cytoplasmic cell T cell, and MSC molecule 2 always presents its antigen to helper cell. This is the rule. Just remember these things, and also remember the binding site. 1, 3, 1, 2, 3, 4 is enough for your understanding. Fourth point is that CD8, always MSC molecule 1 binds with the CD8 receptor on the surface of the T cell, and MSC molecule 2 always binds with the CD4 receptor, which is there on the surface of the Th cell, means helper cell. This is the rule. For your understanding, 4 points are enough for your PG study. Okay, and you have another important thing with regards to the MSC molecule. Just wait for a minute. That is endogenous and exogenous pathway presentation. What we have talked about earlier in the earlier. Always MSC molecule, always antigens. Endogenous antigens are presented by the MSC molecule 1, what we have talked earlier. And always exogenous antigens, okay, are presented by the MSC molecule 2. This is the pathway. Exactly, the MSC molecule helps in the presenting of endogenous how these MSC molecules are play a key role in the presenting of the antigens in the endogenous pathway and exogenous pathway. First, we go through with the endogenous pathway. And endogenous antigen is degraded by proteasome. Okay, the proteasome is a molecule. It degrades the endogenous antigen, and peptide is transported to rough endoplasmic reticulum via TAP. TAP is the transporter, and this rough endoplasmic reticulum where, contains different types of protein molecules. There is called reticulin and tapsin, tetapsin, tepacin. Different types of molecules are there in the endoplasmic reticulum, and this endoplasmic reticulum also contains MSC molecule 1 and 2 in inactive stage, and different different, not in collectively, but it is there, but not in active form. And class M1 MSC molecule alpha chain binds with colnexin, then beta microglobulin, colnexin dissociated, calretirin. Don't bother about this pathway. Just remember thing, that is endogenous antigen can be will be presented by the MSC molecule 1. Just remember this point only. Class 1 MSC peptide is transported from a rough endoplasmic reticulum to Golgi complex, and where they exactly processed and presented with the expressed on the uh, surface of the plasma membrane, and Golgi complex to plasma membrane. This is the pathway what exactly happening in the presenting of the antigen, exactly endogenous antigen. First of all, endogenous antigen coming to the cell. Uh, Antigens are processed by the proteosomes, and in the rough endoplasmic reticulum, it connected with the MSC molecule, and this MSC molecule, along with the antigen, secreted to the Golgi complex. Golgi complex receive the complex of the antigen and MSC molecule, and it's secreted to the plasma membrane. Okay, this is the simple pathway. And coming to the exogenous pathway, and exogenous antigen, when the exogenous antigen come into contact with the MSC molecule, and before the entering to the exogenous antigen, the clip the clip is inactivated form. Clip is covered the active site of the attachment site of the antigen. So after entering to the antigen, this clip will be digested after entering to the antigen into the cell, and HLA-DM mediates exchange of the clip for the antigen peptide, and after the removing of the clip, the complete and attachment of the antigen, the MSC molecule is there, and after binding with the antigens, it secretes on the surface of the plasma membrane, and or it transported to this plasma membrane. This is simple pathway. Invariant chain is degraded, leaving the clip fragment. And, this is the clip fragment means inactivated form. And after entering to the exogenous antigen, endogenous means it is synthesized in the cell. Exogenous means antigen already produced or synthesized outside the cell. In case of exogenous, antigens are produced in the outside of the cell. Okay. In the endogenous means antigens are produced in the cell. Okay. In the metabolic pathway of the bacteria, metabolism of the virus will produces the endogenous antigens. Okay. Some other antigens produced outside the cell, those are called as exogenous antigens. For both antigens, these two MSC molecules are very play a very important role to present these antigens to the T cells. Always endogenous antigens presented by the MSC molecule 1 to T cytotoxic cell. That is the rule. Always exogenous antigens are presented by the MSC molecule 2 to the T helper cell. That is the rule. Enough. Okay. This is about the endogenous presentation, antigen pathways for the antigen presentation. So, simply, this is about MSC molecule. Okay. If you have any doubts, you can ask about MSC molecule. Any doubts, someone? Hello.
Sir, please explain second point and third point in exogenous pathway class to MHC.
Second point and third point.
Yes, sir.
So it's okay, but don't bother about these points. Just remember, in the exogenous pathway, antigens are presented with the help of MSC2. That is enough. That is simple point. Enough for your MSC purpose. Exogenous antigens always presented by the MSC molecule 2. For your understanding, and once again I am telling you that class 2 MSC alpha and beta bind invariant chain blocking the binding of endogenous antigen. The MSC complex is routed through Golgi to endocyte. That is simple. They formed after the formation of MSC molecule. It packed with the Golgi complex. Okay, it is transported into the Golgi complex. Invariant chain is degraded. Okay, this chain is degraded, leaving the clip fragment. Okay, this after the chain, the invariant chain is there now. This invariant chain after getting transported into the complex, uh, Golgi complex, this invariant chain will be degraded and leave some small fragment that will call as clip after the digest invariant chain. Okay, finally this clip also be digested completely after getting interacted with the endogenous, exogenous antigen. Whenever these exogenous antigens come and contact with the MSC molecule, this small fragment of invariant chain also be removed from the MSC molecule, where exactly antigen binds. You simply just remember about the antigen presentation. Always exogenous antigen present to the pH molecule with the help of class 2 MSC molecule. Always endogenous antigens are present with the present to the TC cell. Okay, that means T lymphocyte cell, T cytotoxic cell with the help of MHC molecule 1. That is enough. Understand, Amma?
Yes, sir. Thank you. Understood, sir.
Okay. Yes. So this is about a brief about MSC molecule. Just after this class, I would like to talk about complement system. Complement system, page number 107. You can see your book. That is 107 page. What is complement system? The complement system is common component of normal serum. Those complement means complement in the sense complement proteins. Okay, there are different types of proteins are there. Those proteins are called as complement proteins, which are helping in the formation of membrane attacking complex, which are helps to the elimination of the different types of infections and different types of antigens from antigen antibody complexes from the circulation or from the tissue spaces. Those proteins, some of the proteins are there to which helps to the our immune system. Okay, those types of proteins are called as complement proteins. Okay, so and complement is the common component of normal serum and major effector for the antigen antibody reaction. And complement can be regarded as an innate defense mechanism. This complement system can be regarded as innate defense mechanism. I have already told you that there are different barriers are there. After barriers, inflammation, phagocytosis, complement system also uh, comes under innate branch of immunity. So antibody can also be involved in its activation. Though this complement system comes under innate immunity, but antibodies are also a role of the antibodies are also they be there in this complement activation and provide the actual protection from the response while infection of antibodies and antigens determines the specificity of response. Antibodies finger the target and complement destroy it. Okay, this is a very very important sentence. Antibodies finger the target. Antibodies finger the target means antibodies goes and binds with the infected area antigens. The surface antigens you may different types of antigens. Okay, those antigens are recognized with the antibodies, and these antibodies goes and binds with the antigens, and that is the meaning of fingering. And these antibodies fingers the target. After the attachment of the antibodies, the antibody Fc region complement protein binding site also be there. So after the attachment to the antigen, antibody to the antigen, these complement proteins get interacted with the attached with the Fc region. So that facilitates the destroying, thus facilitates the destruction or killing of the pathogen and parasite. That may be infection site. That is the infection site. Okay, that means antibodies finger the target site and complement destroy it. Okay, that is the exact meaning of that sentence. And complement components are normal serum proteins. These components constitute the phi of the serum globulin function fraction and form 20 different types of proteins are there. How many proteins? Almost all 20 different types of proteins are there, and which are also some of them also among the 20, some of them are also acts as the enzymes. Okay, some of them are control molecules, some of them are structural proteins with no enzyme activity, and among the 20 proteins, Some of them are act as the enzymes. Some of them are control the molecules, other molecule activities. Some of them are also acts as a structural proteins with no enzyme activity. These three types of characteristics are there for the complement proteins, and of these 20, among these 20, about 16 complement proteins are biologically important. Among the 20, 16 proteins are biologically important, and can be designated as letters. Okay, C, letter C. Okay, all complement proteins can be designated as C. That means complement protein, and numbered also for the each and every protein. We can scientists given has given names, numbers also, that C1, C2, C3, etc., etc. Complement one contains three subunits. Okay, among the proteins, only one complement protein one consists three subunits. Very very important. C1Q, C1R, C1S. Okay, these are the three sites in the C1 protein are having the major role to stimulate the uh, classical pathway. To stimulate the classical pathway, these are the very very important subunits in the class 1. Okay, now we can also having the serum contains the factors. Okay, some of the factors also there along with the proteins. Some of the factors also playing a major role in the complement uh, system. Those are designated as B factor, D factor, P factor, H factor, and I factor. These are the factors which helps the complement proteins to making the it function well. The complements are synthesized in various sites throughout the body. Okay, especially in the liver. Okay, in the liver there some of the proteins are produced. C3, C6, C8, B factor, and other intestinal epithelium C2, C3, C4, C5, factor P, D, and I are synthesized in the site, and uh, and different also different types of proteins and factors also produced in the macrophages. You know, you have all those information in your book, and this system, the main function or goal of the complement protein system is that lysis of cells. Okay, to coming to the function, before knowing to the different types of pathways, you must know about the function of complement system. First complement system function is that lysis of cells of bacteria, viruses. Okay, those are having the uh, bacteria. Those cells are having the bacteria or virus, means infected cell. Lysis of infected cell is a major function of the complement protein or complement system. And second function is that opsonization, means it promotes the phagocytosis of pathogen or antigens. Okay, it attracts the phagocytic cells to the site. Okay, uh, and another function is that activation of inflammatory response is one of the another function of the complement system. And other function is that clearance of immune complexes. Wherever the immune complexes are there, those are the harmful for our system, and prolonged storage of the complement binding immune complexes may lead to the hazardous effect on the system, so that may must be eliminated from our system. So majorly the complement system play a key role in the elimination of the antigen antibody complexes from our system. Okay, that is the major function of complement system. One is that lysis of infected cell, opsonization. Oh, that means promote the phagocytosis, activation of the inflammatory response, and clearance of immune complexes. That means antibody antigen complex. These are the functions, and and we will discuss about lysis of cells. and all these complement proteins are actively involving in the making the pore on the infected cell. Okay, it makes the pore on the infected cell by different mechanisms. Uh, we have three different pathways to make a pore on the cell, and one is that classical pathway, and which leads to the final goal is that making a pore on the infected cell or the bacteria cell surface or the different types of parasites cell surface. It makes the pore on the cell by the different pathways. One is the classical pathway, second one is the alternate pathway, third one is lectin pathway. Coming to the classical pathway, all the pathways are having the proteins. Okay, the complement proteins. With the help of this complement protein, different complement proteins, it makes the pore. How we will see. And in the classical pathway, antigen antibody complex is play a key role and activates the C1 molecule. This is very very important. I will show one image for you to better understand. What is where exactly is it? Yes, yes. In the classical pathway, what happening that antigen antibody complexes, wherever these complexes are there, this C1 molecule goes and binds with the antigen antibody complex. In the C1 molecule, we have it contains three subunits. Where exactly this uh, C1 molecule binds with the antibody? With the help of CQ subunit. CQ subunit goes and binds with the antibody. It activates the entire C1 molecule. Okay, other subunits of the C1 protein. Whenever the antibody antigen complexes are there in our body, this C1 molecule goes and binds with the antibody, especially with the help of CQ subunit. And this binding facilitates the further activation of C1R and C1R2. Okay, and this activation may further activate the C1S. Okay, which is one of the another subunit of the C1 molecule, and C1S. Whenever the C1S get activated, it also activates the C4 and C2. It cleaves the C4 and C2 molecules. Okay, this C4 and C2 molecules will be cleaved in the presence of the C1S, activated C1S. Okay. This is the initial step. What happening in the initial step? Whenever the antigen antibody complexes are there in our body, and this C1 molecule goes and binds with antibody. Okay, especially with the help of CQ subunit. CQ subunit goes and binds with antibody, and this binding facilitates or leads to the further activation of another subunits of C1 molecule. Those are C1R, C1S, and activated C1S cleaves the C4 and C2 molecule, and this C4 and C2 molecule get activated in the presence of C1S and gives C4A, C4B subunits, C2A, C2B subunits. Okay, and with the combination of C4B, C2A, and it acts as a C3 convertase. Okay, after the cleaving of the C4 and C2 and C4 subunit B, C2 subunit B or A binds with together and acts as a enzyme. Okay, these subunits, whenever these subunits get interacted with each other, it acts as a enzyme. Okay, that is convertase. Okay, in the presence of this convertase, C3 molecules will be cleaved in the presence of this enzyme, that is C3 convertase, C4B, C3A, CA, not B, this is not B, A exactly. And in the presence of C3 convertase, C3 cleave into the C3A and C3B. And after the cleaving, C4B, C2A binds with the C3B molecule and also acts as a C5 convertase. Okay, it also acts as a C5 convertase after combining with the C3B subunit with the C3 convertase, it acts as a C5 convertase. And in the presence of C5 convertase, C5 complement protein cleaves cleaved into the C5A, C5B, and C5B goes and binds with the C6 molecule, C7 molecule, C8 molecule, and this further C9 molecule combined with this entire unit and acts as a membrane attack complex. And finally, this C5B 6, 7, 8, 9 combinedly acts as a membrane attack complex. Whenever all these subunits and proteins bind with each other, this it makes a pore on the membrane of the infected cell or infected cell or pathogen or parasite. This is the main goal of the complement system, making a pore on the infected cell on the surface of foreign body, maybe bacteria or parasite, protozoan parasite, element parasite. This is the major goal. This uh, making a pore on the body of the foreign by the three pathways. This is one pathway, and this is second pathway. This is third. All the pathways are the junction of these three pathways are C3 convertase. C3 convertase is the junction of this among the three pathways. In all pathways, C3 convertase is must is the one of the steps is the junction for the all pathways. C3 making a C3 pathways is different in the different pathways. In the first pathway, antigen antibody facilitates the formation of the C3 convertase, and in the second pathway, binding of uh, MBL. MBL means monon binding lectin. Okay, monon binding lectin is a carbohydrate molecule on the target cell surface. Okay, target cell surface contains it monon binding lectin molecule. It is one of the carbohydrate molecule. It is there on the target side. The target means infected cell or pathogen. Okay, okay. Binding of MBL or what is this? Phycolin. Okay, to pathogen surface. Pathogen surface contains this MBL molecule. Okay, MBL molecule contains there on the surface of the pathogen, and this MBL molecule recognized by the C1 protein, C1q binds with the MBL. Okay, that may leads to the formation of C3 convertase in the second pathway, that is lactin pathway. Coming to the alternative pathway, in that pathway also C3 making of C3 convertase is necessary. All these pathways leads to this formation of C3 convertase. What happens in the alternative pathway? In the alternative pathway, spontaneous breakdown of C3 to C3A and C3B that may leads to the formation of C3 convertase. Okay, spontaneous uh, breakdown. Okay, is one of the reason, and also it is the independent. Independent means to generate C5B antibody independent. No need not to participate need not to participate the role of the antibody. The antibody not necessary to generate the C3 convertase in the in these two pathways. Okay, for these two pathways, no need to contact with the antibody antigen to the antigen. So directly it provokes the C1 molecule. It binds with the C1 molecule in this pathway and leads to the C3 convertase. Whenever the C3 convertases are produced, that is a very important role and play a key role in the making the pore formation. And C3 convertase converts the cleaves the C3 molecule, which is very important for the formation of C5 convertase, and C3b with the C3 convertase and acts as a C5 convertase and cleaves the C5 molecule, and it is a very important convertase enzyme. And C5b, whenever the C5b is free in the system or circulatory system, it attracts the 6, 7, 8 and binding sites for the 6 and 7, 8 is there on the C5b, and the combination of the C5b, 6, 7, 8 may attract the ninth complement protein, may leads to the membrane attack complex. This is how the uh, complement protein complement protein makes a play a key role in the formation of membrane attack complex, which helps to the uh, elimination of the different types of pathogens and parasites. Okay, this is membrane attacking complex. This is the infected cell, and these are the 5, 6, 7, 8, 9 molecules. Okay, this is the classical pathway from here to here. Plus all the pathways leads to the formation of the C3 convertase. In all the pathways, C3 convertase is the common point or junction. Okay, 1, 2, 3 pathways leads to the conversion of the uh, C3 molecule, or the cleavage of the C3 molecule. That is come from C3 convertase is the junction between all the three pathways. After the C3 convertase, C3 molecule convert into the splits into the two subunits, C3A, C3B. C3B go and bind with the molecule and gives the C5 convert into the C5 convertase, and C5 convertase cleaves the C5 molecule, and C5 molecule cleaves into C5B, C5A, and this C5B has a binding site for 6, 7, 8, and these whenever these molecule combines, and C39 also attract and binds with these complex and makes as a form as a membrane attacking complex. Okay, this is about complement protein, and complement um subunits. The main important role of this subunit is that coming to the C1, especially CQ binds with the antibody. It is there in this information is there in your page number 111. In the 111 page, you have full information. CQ molecule, function of CQ molecule binds to the antibody bound to a pathogen, and thus allowing autoactivation. Bind to antibody is a classical pathway, and bound to pathogen is a alternate pathway. Okay, and C1R cleaves C1 to activate protease, and C1S cleaves C4 and C2, and that is the function of the C1 molecule. Coming to the C2, C2A active enzyme of the classical pathway, C3, C5 convertase, cleaves C3 and C5. Okay, it is a part of uh, convertases. Okay, C3 and C5 convertase. Uh, it is a part of one of the part in the uh, convertase enzyme, and C2B precursor for the various vasoactive C2 kinase. Don't bother about this C2B. Just remember C2A role in the complement protein. And coming to the C3B, C3B binds to pathogen surface and acts as a opsonin. And C3B role is opsonization. Very, very important. Initiates amplification via alternate pathway, binds C5 for cleavage by C2B. Okay, and C3A role is the peptide mediator of inflammation. Wherever the inflammation reactions are there, this inflammation is this C3A may leads to the inflammatory reaction. And coming to the C4 complement protein, and C4B covalently bind with the pathogen, and opsonization is the one of the function for the C4B. Coming to the C4A, peptide mediator for inflammation, anaphylatoxin. Okay, acts as an anaphylatoxin, and inflammation is the function of C4A. And almost all, I have covered all the things in brief. The complement system. Just read the points and read the book. If you have any doubts, you can ask in the next class, subsequent class. Okay. Okay, Namu.
Okay, sir.
Just read once. If you have any doubts, you can ask in the next class. Up to second unit, we have discussed second block, not unit. Up to second block, we have discussed briefly. In the next class, we will be talking about vaccinology. Okay, vaccinology. Okay,
Okay, sir.
okay, all right.
Thank you, sir.
Okay, can I leave
Thank you, sir.
now? Okay, okay, bye.
Yes, sir. Thank you, sir.
Okay.